For instance, I know that you've heard from probably your early high school biology how glucose is used by the body to be broken down into generating ATP for our cells. Fructose, you can find these in a lot of syrups that we eat. These are a 5-carbon ring structure, so they lack 1 carbon in comparison to glucose, which has 6 carbons. Sucrose is a disaccharide, and one thing to note is that fructose and glucose are both monosaccharides. Sucrose is a disaccharide, which means it's 2 sugar molecules joined together. So essentially, you could have 2 molecules of glucose that are connected via a chemical bond, and those molecules will make up a disaccharide, which will be in the form of sucrose. Same thing for lactose and maltose. Maltose is a large polysaccharide, same as starch, and cellulose and glycogen. So the examples that's given, you kind of have a monosaccharide here, monosaccharide for fructose, and then disaccharide for sucrose, disaccharide for lactose, and then all these other ones are very large, complex carbohydrate molecules. So they just get bigger in complexity, but our body needs to use all of them to survive. So different categories of carbohydrates is just like what I said: monosaccharides, disaccharides, and then polysaccharides. Here's that monosaccharide glucose molecule I've been telling you about, the C6H12O6. And then when you have 2 of these molecules joined together, you essentially get a disaccharide, which could be an example could be sucrose. And then we have polysaccharides, which are many monosaccharides all joined together. We have 2 disaccharides that make up a polysaccharide. So the bigger and more complex the carbohydrate is, the more energy your body will need in order to break apart these chemical bonds and extract the energy from those bonds to drive your metabolic processes. Outside of our body, microbial organisms, they have to figure out ways to break down these large polysaccharides. So think of something like a potato. A potato is about 99% nothing but starch. And those starch molecules, in order for a bacteria or fungus or us to extract nutrients from the chemical elements that make up those starches in the potato, those molecular structures have to be broken down. So we're talking about hundreds of chains of polysaccharides all throughout the potato, which requires a lot of energy to utilize in order to break these apart into smaller disaccharides and break them further apart into monosaccharides, and then splitting this glucose molecule directly in half so that way it can be used in the process of cell respiration or be used in the process of fermentation. So those are saccharides. Now the monosaccharides of the group, they're going to be the most simplest of the carbohydrates. Mono means 1. Again, you have 1 single particular structure, and glucose is the primary example that we use to kind of comprehend monosaccharide. There's a couple different configurations that there are. There's alpha and then there's beta ring configurations. I'm not really too concerned with you understanding the differences between alpha and beta ring, but just know that the C6H12O6, as we talked about, can either be in linear form or it can be in the cyclic form, which would be the ring structure. So here is glucose. The molecules in this glucose, the numbers are exactly the same as these that are all listed over here. But what's different, what makes glucose different from another molecule known as galactose and another molecule as fructose, is the way that the molecule is arranged in terms of the chemical elements that make it up. So this has a bunch of hydrocarbons in it. So we see carbons, carbons as our primary backbone of the molecular structure. You see OH groups, which is oxygen. That's also bound to carbon, but also bound to a hydrogen. And then we have a double bond to O, which indicates that we have glucose, or a type of OS molecules, like sucrose, lactose, or any of the O's indicates a carbohydrate. So this double bond to O is kind of a key signature that what you're looking at is a carbohydrate. So glucose is the primary example, but in nature or in a laboratory setting, If there is manipulation of this structure, then you can end up with a different compound. So for instance, notice galactose and glucose are exactly identical as you look where all of the chemical elements are lined up, but where the red is highlighted, you have a difference in structure. So here we have the OH having a share pair bond with the electrons that are on the carbon atom's right hand side. So that's our left as we're looking at the picture. But we have a bonding of a share pair of the electrons that takes place here versus the other side where we simply just have a hydrogen. So in the glucose molecule, that's flipped the other way. So and don't ask me why this is the case. It just happens in organic chemistry. But once these the positioning of the chemical elements shifts or changes, you end up with a different molecular structure, very slightly from the original molecular structure, and so that will develop a new molecule. So that changes glucose into a new chemical compound called galactose, which is also utilized in the body as well as in cells in the process of cell respiration. Fructose is also another C6H12O6 molecule. And so this configuration here that you can see that the double bond to oxygen has been altered in this molecular structure. That's the primary difference. The C double bound to OH, the third carbon, is in the same place as the glucose. So this area here that's outlined in red is the only key feature on here that has been manipulated or been changed to give us a new chemical molecule. So you have the second carbon in this chain of carbons that's double bound to the oxygen. And then you have essentially what would be the first carbon for both galactose and glucose as being the head of this fructose molecular structure. So the structure of how these chemical elements are put together dictates what type of chemical compound is going to be developed. But what is similar here is the fact that all of these have C6H12O6. They're just different in structure. Okay, so glucose, as I said already, I think I keep hitting this concept home, is that it is a major energy source for the body because it it contains 6 carbons. And so what the body does is what your cells will do is it will break the bonds that hold this molecular structure together and split this molecule into 2 chains, 2 3-carbon chains. So you can split it probably, on the chain it's easier to see, so you could split it right in half, maybe break the bond. What is 1, 2, 3, 4, 5, 6. So maybe the bonds between, if this is, here's carbon 1, carbon 2. So you break the bonds between carbon 3 and 4, and that'll give you your 2 molecules of what we call pyruvic acid, and or 2 molecules of pyruvate, and then it gets converted to pyruvic acid in the cell respiration cycle. But in here, the cyclic cycle, the cell would also need to cut probably between these 2 carbon chains here, or carbon bond where the electrons are here, between C3, which is carbon 3, and carbon 4. So you break this particular bond, but you also have to break the bond between the carbon and the oxygen to enable the, at least your cells do this, to enable the conversion of your 2 3-carbon chain molecules into pyruvic acid, which then feeds into the Krebs cycle. And once it feeds through the Krebs cycle, it'll go through the electron transport chain, and then you'll generate ATP for your cells. So all of this takes process only because we utilize glucose in this form or fashion, and that's, again, the primary energy source for the body. So as we're going along learning about the different types of carbohydrates, we'll go back to talking about disaccharides. Di means two, saccharides refers to carbohydrates or sugars. And with these particular group of carbohydrates, you essentially again have two monosaccharides that are joined together. When these monosaccharides are joined together, that happens due to the process of dehydration synthesis, where essentially you have the removal of a water molecule that allows the two monosaccharides to join together. So if you look here on this glucose molecule and this beta fructose, they're joined together, are bound together by an oxygen that's present. Now, this oxygen was only able to bind these two molecules together if glucose and beta fructose were able to lose some hydrogens that would have been affixed to carbon 2 and carbon 1. So this carbon on this glucose molecule was originally bound to another carbon here at this corner, an oxygen, and then a hydrogen. And then for stability purposes, it would have been bound to a hydrogen if it was just a simple monosaccharide. Well, that particular hydrogen has been lost out into the environment, and that's opened up a valence electron pair sharing location where oxygen can come in and share the electron here that carbon still has to be shared. Same thing happens on this carbon here, which is originally bound to carbon. It's bound to an oxygen and it's bound to another carbon. So that would leave a hydrogen that it would have been bound to. So through the process of dehydration synthesis, we're able to generate larger molecules. So this is how monosaccharides can become disaccharides and how disaccharides can become polysaccharides. Now, in order to break apart disaccharide molecules, we use the process of hydrolysis reaction. And the only thing that happens here is simply that water molecule is going to hit where that oxygen bond was originally, and it's going to break that apart. And so once the glucose molecule separates from the fructose molecule, that opens up, again, locations that can be shared between other chemical elements. And so what happens is the water molecule comes in, the water molecule gets split, so then you end up with two hydrogens that are free floating, and then you have an oxygen. Well, one oxygen is going to stay bound to the carbon that it was split from, or between the two monosaccharide molecules it was split from. So oxygen is going to stay, and then the other oxygen will bind with a hydrogen, and so you have an OH group that gets applied to this glucose molecule now. So this water molecule is able to break apart disaccharides and polysaccharides using this fashion. Now, this entire process can be expedited through the use of an enzyme, and the enzyme that's involved in this process is called sucrase. Anytime you see ASE at the end of a chemical, typically that means that you have some sort of enzyme that's present. So sucrase is very specific, and it will only break apart the smaller monosaccharides of a sucrose molecule. So this is our sucrose molecule. Both sucrase and water hit this oxygen bond right here, and then once that hit, it breaks apart the two monosaccharides that make up the disaccharide, giving us now glucose and a fructose molecule. So to recap, if you want to build disaccharides or polysaccharides, you're going to use the process of dehydration synthesis. I think I was on slide 20 and 22. You're going to use the process of dehydration synthesis, okay, in order to build molecules. To destroy molecules, we use the process of hydrolysis, which is assisted by the use of enzymes. Okay, and then here's a schematogram that outlines that for you. So again, we have hydrolysis, and then we have dehydration synthesis, where you have the production of larger molecules. With hydrolysis, you have the destruction or breaking apart of small molecules. Okay, So we haven't really talked too much about the cell structure of a bacterial cell, and we will delve more into that in upcoming lectures. But since we're talking about biochemistry, we want to kind of understand the context of why this is important, especially to this course.